Hello, I'm Elia Mascolo, and welcome to my brief overview of my poster, Molecular Information Theory of Composite Sequence Motifs. If we think of a transcription factor searching for its targets on a genome, we can use information theory to measure in bits how much information is required for this task to be accomplished. This quantity, which only depends on the genome size in base pairs, G, and the number of target sites, gamma, was called R frequency. But as biologists, we also understand how the information received by the transcription factor is encoded. It's encoded as nucleotide sequence conservation in the target sites. So when we look at the motif, we can see that each position provides up to 2 bits of information, and the total information was called R sequence. A beautiful result from this theory that was developed in the 80s is that the values of R sequence that we observe tend to be approximately equal to the predicted values of our frequencies. And this is true for transcription factors as well as other DNA binding proteins. What we do in this work is to generalize this well-established theory so that we can use it to talk about cases where regulation happens through the recognition of composite motifs. And by that, I mean a combination of several sequence patterns at a certain distance from each other. And so the distance between sequence patterns can be more or less conserved, providing more or less information to the regulator. And the amount of information provided by those spacers is what we call R spacer. When we generalize this framework, we have a new parameter n, which is the number of sequence patterns in the composite motifs, and what we find is that there are certain bounds within which there's some freedom as for how much information could be encoded in as R spacer versus R sequence, as far as the sum of the two things matches a certain quantity that the theory predicts. We discuss the regulators that have to recognize these composite motifs from the statistical mechanics point of view. And we expect the mechanical properties of these regulators to be optimized in a way that matches the properties of the targets. Specifically, the protein flexibility, the optimal protein flexibility will depend on the variability of the spacer in the recognized targets. We also discuss these systems from the thermodynamics point of view. In particular, we differentiate between recruitment model, where the components that have a DNA binding domain search the genome independently, and after binding, if they are at the right distance from each other, they can interact in a way that can modulate transcription. And we compare that with the pre-recruitment case, which is when the multimeric regulator is first assembled, and then as a whole, it searches for its targets in the genome. And we discuss under which circumstances one strategy would be more thermodynamically efficient than the other. We ran evolutionary simulations where initially random genome sequences evolve transcriptional regulatory systems from scratch, And we use those simulations to validate empirically the theoretical results that we obtained. We also use those simulations to show that there is a strong selective pressure towards the optimization of the mechanical properties of the regulators, so that they are optimized for their search according to our biophysical results. We also show that when mutations are dominated statistically by base substitutions, there is an advantage in prioritizing our spacer over our sequence, meaning that spacer size will be maximally conserved, and that because this strategy guarantees mutational robustness. But if small insertions and deletions are too frequent, then there can be an advantage in using a strategy that encodes less information as our spacer and more as our sequence. Thank you for listening. I am looking for feedback and networking opportunities. So if you are into theoretical biology, please reach out.